In this practical, we are going to amplify a fragment of DNA of a parasite in a human blood sample. The technique that we are going to use today is known as polymerase chain reaction, which we commonly known as PCR. PCR is one of the most reliable methods to diagnose an infectious disease. It is being used not only for medical diagnostics but in many fields in science, including forensic science, DNA sequencing, research in science and medicine. PCR is a chemical method to copy or amplify target fragment of DNA inside a small glass tube. Thus, it is also known as an in vitro method of DNA synthesis. It synthesizes a billion copies of identical DNA fragments of a target DNA molecule. This is similar to the natural DNA replication process that happens in your body cells. Amplification process to happen, the following 6 components should be contained inside the reaction mixture. The PCR process involved these 3 major steps. During denaturation, double-stranded DNA melts into 2 single-stranded molecules and each act as template for new strands to be formed. During the annealing step, primers bind with their complementary sequence on the template DNA, marking the area of the template DNA to be copied. During the extension, it adds nucleotides to the ends of the annealed primers, thereby synthesizing the new molecules during the reaction. This is how DNA replication synthesizes a billion copies inside a small glass tube during PCR. This is the area we need to copy and amplify within the template DNA molecule. In order to copy this region, PCR needs 3 other ingredients: 4 types of nucleotides, DNA primers, and the enzyme Taq DNA polymerase. Primers are single-stranded short DNA fragments which are having complementary sequence to the ends of the target DNA region. The DNA polymerase is an enzyme extracted from thermophilic bacteria known as Thermus aquaticus. Since the enzyme is heat stable, it can withstand the intense heat during the PCR reaction. PCR consists about 30 to 35 cycles. At the first cycle, it begins with heating the reaction mixture up to 95 degrees centigrade. The heat denatured the DNA template, breaking the hydrogen bonds and separating 2 strands apart. After denaturation step, the temperature is reduced to 60 degrees centigrade. At that temperature, the primers will find their complementary sequence on the template DNA and binds to it. This step is known as primer annealing. This annealing follows the base pairing rules. A adenine binds with T and the cytosine C pairs with the guanine G. In the next step, temperature is gradually increasing up to 72 degrees centigrade. Taq DNA polymerase works optimally at this particular temperature and start polymerizing by adding corresponding nucleotides to the 3' end of each primer. At the end of this cycle, there are 2 identical double-stranded DNA molecules of target DNA is created. After the second cycle, 4 identical copies of the target DNA is created. After the third, there are 8 copies of double-stranded DNA is synthesized. As the number of cycles increase, the number of nucleotides and molecules synthesized within the reaction mixture increases exponentially generating billions of copies of identical DNA copies at the end.
Now we are inside the PCR lab and about to diagnose the presence of a DNA of a parasite in a human blood sample. The reaction is set up inside a special chamber which is known as a PCR hood. Since PCR is an extremely sensitive reaction, even to trace amounts of DNA, this would help us to prevent any unwanted contaminant DNA to be amplified during the reaction. There are several instruments and reagents need to set up a PCR reaction. Among those, this is one of the most important equipments that we are using, a micropipette. So this help us to dispense micro volumes of liquid or reagents into our PCR mixture. In order to dispense micro volumes, we are attaching the desired size of pipette tips with the micropipette. This micro tip contains a special barrier right in the middle of the tip barrel. So this type of a barrier tip prevents any aerosols that are going to be generated while aspirating the liquid in, getting into the barrel of the tip. So using this type of a barrier tip is very important for us to prevent sample cross-contamination when we are setting up of the PCR. You also need a ice bucket or a cold block like this to retain your chemicals while setting up of the PCR. So this cold conditions is very important for a successful PCR at the end. Let's see what are the reagents need to set up the PCR reaction. The first chemical that we are adding into our reaction is the PCR buffer. PCR buffer provide optimal solution conditions for the other reagents to work. It is normally added into the mixture at first. Next chemical that we are adding is magnesium chloride. Magnesium chloride helps to anneal the PCR enzyme to the DNA strands. The next goes into the reaction is the primer mix. This primer mix contains equimolar concentration of forward and reverse primers. Primer is a piece of DNA which corresponds to the pieces of DNA that you are trying to amplify during the reaction. The next thing that we are going to add into our reaction mixture is dinucleotide triphosphate mixture, or commonly known as DNTPs. This contains a mixture of 4 nucleotides that needs to synthesize the new DNA molecules during the amplification process. The final component that we are adding into the mixture is the enzyme. This enzyme is known as the Taq-DNA polymerase. Taq-DNA polymerase is a heat-stable enzyme which has the capability of polymerizing nucleotides one by one sequentially when the reaction happens inside the tube. The final volume of the master mix is adjusted by adding double distilled water into the master mix. The final component that goes into the reaction is the extracted DNA. Extracted DNA in PCR reactions are also known as template DNA. In the exercise that we are doing today, in this particular DNA extraction contains not only the pathogenic DNA but also the human DNA, because of the fact that this was extracted from a blood sample of a human. To set up the PCR, we are using two types of tubes. The first one is known as the microcentrifuge tube, which has a total volume of 1.5 milliliters. The second tube is the PCR tube, where the reaction is taking place. The total volume of this tube is 200 microliters. During the entire process of setting up the PCR, you need to wear gloves. It is very important to wear gloves because that will prevent any unwanted contaminations going into our reaction mixtures. Once the samples are prepared, they are placed in a machine like this. This machine is known as the thermal cycler, or commonly known as a PCR machine. This machine can be pre-programmed to retain certain temperature to be held at a certain period of time. Once the tubes are inside, the chemicals will gradually amplify the DNA template that we have inserted in. Now we are preparing the master mix inside the PCR hood. The first chemical that I am adding into the master mixture is the PCR buffer. So the buffer was added into the tube, and the second chemical that we are adding in is the magnesium chloride. Between reagents, make sure that you always change the pipette tip so you don't contaminate in between the reagent. So the magnesium chloride mixture is also added. The third chemical that goes in is the T.S.P mix.
Then the primer mix goes into the reaction mixture.
The next one is the Taq DNA polymerase enzyme, which is going to be used as the main enzyme for the polymerization activity during the reaction. And each time you have noticed that I have adjusted the volume of the micropipetter according to the volume dispensed. Finally, I am adding the total volume of the master mix with the desired volume of the double distilled water. After adding all the chemicals together, the mixture has to be generously mixed, and then this is ready to be dispensed into our PCR tubes. These are the PCR tubes. The PCR tubes, as you can see, have been pre-labeled beforehand to ensure that you are adding the correct sample to the correct tube. So the total volume that I am dispensing into each PCR tube is 18 microliters. In our reaction, the first tube goes with the extracted DNA sample, and the second PCR tube is for the negative control of the reaction, and the third one for the positive control reaction. When setting up the PCR, it is important not only to run the sample itself, but also a negative control, a master mix without the template DNA, and also positive control with a known sample which is already the targeted DNA sample inside the extract itself. Now we are ready to add DNA template to our PCR tubes. Into the first tube, we are adding our extracted DNA sample. Then for the second tube, we are adding an equal amount of distilled water instead of a extracted sample, because the second tube is the negative control of our assay. To the third tube, we are adding a extracted sample of a non-positive, because our third reaction is the positive control. Now our samples are ready to go into the thermal cycle. So these are our three reactions that are going to run in this machine. Now our reactions are inside the machine. Now we have to select the desired method and run the PCR machine accordingly. Now our run has initiated. As you can see in this protocol, there are three main stages: stage 1, stage 2 and stage 3. Stage 1 is for the initial denaturation step. As you can see, the temperature is 94 degrees centigrade and it holds up for 2 minutes. In the second stage, you can see the initial denaturation step, where during this step, that allows complete denaturation of our template DNA inside the reaction. And in the second step, during stage 2, the primers in the mixture going to be attached to our template once the tube is cooled at 60 degrees centigrade. The third step at stage 2 heats up the tube again up to 72 degrees, where DNA fragments will start amplifying itself by using the DNA polymerase enzyme. Finally, at stage 3, the temperature is 72 degrees again, and it will hold up to a period of 5 minutes to the final extension step. If you look carefully, during stage 2, these three steps will gonna be alternate or cycled about 35 times again and again. During this cycling process, our DNA template inside our sample tubes gonna be amplified in millions of times. This full copying process will be completed within a period of 2.5 hours of time. Once the cycling is completed, samples can be removed from the machine. Now our PCR is over, and we are taking our amplified DNA samples out of the machine. In order to visualize whether we have obtained any amplification or not, we need to run these samples in a agarose gel by using agarose gel electrophoresis procedure.